Okay guys, first thing I want to make a very large disclaimer that I have no knowledge of geology whatsoever. I'm not sure why I'm in this session, but I'm assuming it's appropriate. And I would like to request exemption from any questions regarding Jamie's claims about iron and it having nothing to, ferric to ferrous having nothing to do with oxidation in the atmosphere. Okay? So what I wanted to do is to talk about iron, the soluble form, Fe2+, and its role of biochemistry in early Earth. So if we look at the history of biology, not geology, history of biology, as you see here, we see this large tree of life. The tree of life in modern times is represented by three domains: bacteria, archaea, and eukarya. So if we walk back in time along that tree of life, we get to what we call LUCA, last universal common ancestor. We've all heard of that by this point, which we estimate roughly is about 3.9 billion years ago. If we go farther back in time, we get into the squiggly line. And the squiggliness of this line, all that means is that we are likely to have observed a lot of horizontal gene transfer during that time. So even though last universal common ancestor is singular, most likely it's a composite of multiple types of organisms. And what we're really dealing with this far back in time is more like a soup or a sea of different life forms. So what I want to talk about is this point, the RNA world hypothesis, essentially, in which we theorize that there's some kind of primitive catalysis and maybe some metal-assisted assembly back in that RNA world. So the RNA world hypothesis essentially states that before modern biology, which is DNA, RNA, and proteins of our modern times, we had something called the RNA world, in which RNA performed the roles of genetic storage and catalysis, essentially all the functions that protein, DNA, and RNA do together today. And this world is hypothesized to have occurred back in the early Archean eon, about 3.8 billion years ago, very roughly. So what do we know about modern RNA? Well, one thing that we study a lot in our lab, Lauren Williams' lab at Georgia Tech, is the relationship between RNA and divalent cations, especially magnesium. So we know that RNA and magnesium have a very special relationship. And it has to do with this magnesium being a divalent cation, having two charges, and really enjoying the opportunity. Whoops. What did I do? Okay, sorry. And really liking to bond with those phosphates that form the DNA backbone, or the RNA backbone in this case. Oops, I did it again. Sorry about that. And we know that magnesium also has a very special role in the folding of large RNAs. So there are certain, if you have an RNA that's 3,000 bases long, such as what you see in the ribosome, there are certain conformational spaces or certain three-dimensional shapes that RNA cannot reach without the presence of magnesium in solution, just because of its size and its charge density. So we see this kind of special relationship in chelated magnesium motifs, like those found in the ribosome. I believe there are 23 of these magnesium chelation sites in the 23S ribosomal RNA. We'll talk about biology more tomorrow. But just know the ribosome is the thing that makes messenger RNA into proteins in all cells. It's present in all cells, completely ubiquitous. But we see these chelation complexes where we have two magnesiums and then we have multiple phosphates. These are the phosphates on the backbone interacting with the positive charge from the magnesium. And then if we superimpose ribosomes from across the phylogenetic tree of modern life, what we see is that these chelation complexes, this geometry, is conserved among all of life, especially at the center of the ribosome. Oops. I'm going to get the prize for good slide changing this time. So one thing we've done, we have a research scientist in our lab named Anton Petrov. He's a quantum physicist. And so he's looked at in silico, meaning on computer in mathematical space, the relationship between magnesium and RNA. You see here, calcium in RNA and sodium in RNA, trying to identify what is so special about magnesium that these other cations don't have. And so what you can see in these quantum mechanical calculations in mathematical space is that the interaction energy between RNA and magnesium, the interaction energy between those backbone phosphates and magnesium, is much more spontaneous. So interaction energies with a very large negative number are more spontaneous. We don't have to add any energy to them in order to form that structure. But if we look at calcium or sodium, we have a much lower negative number in the case of calcium, or a positive number in the case of sodium, indicating we need to add some energy for that structure to form. Now, what we were interested in looking at is whether some other cations can play the role of magnesium, and we thought we found one. We're pretty sure we found one at this point. So if we look at iron, we put iron in place of magnesium in the same mathematical space, and then we minimize the structure, what we get is the same geometry in the backbone of the RNA with iron as we do with magnesium. We believe that's because the charge density and the size of iron is so similar to magnesium, much more similar than any other cation that we have biologically available. And in the case of iron, actually the interaction energy is a little bit more negative. So the complex formation is a little bit better with iron than with magnesium. So based on this information, what we've hypothesized is that back in the days when the Earth was very anoxic, and I don't think I have to go much into this at this point. We've had a very good overview today. So before photosynthesis and the great oxidation, what we had, as we've already heard, is that the iron in the ocean, the biologically available 2+ form, is probably much greater in concentration than it is today, and would have been much more biologically available. I did want to mention that iron 2+ in the presence of oxygen gives rise to Fenton chemistry, which can be toxic to biology. Oops, and I did also want to show the banded iron formation that we've already seen. So the theory is that as oxygen increased in the atmosphere, we have the interaction with the surface oceans, and the iron precipitated out in the form of these banded iron formations. And this one, I believe, is in Minnesota, courtesy of Clark Johnson. So our hypothesis is that iron, back in the days when the Earth was anoxic, actually played the role in RNA, and played the role in life that magnesium plays today. So we wondered whether we could recreate these early Earth conditions, remove the magnesium, remove the oxygen so that the iron would not eat up the RNA, and see if we could find some kind of activity. See if RNA would do something interesting in the presence of iron. So one of the first things we did is evaluate, like I said before, magnesium is required for RNA to generate certain conformational states. We wanted to see if iron, in the presence of iron, these same conformational states of RNA could be reached. So if you look here along the x-axis, this is essentially, if you imagine this, the 5' end of an RNA, so the beginning of one strand of a molecule. And you follow this along, this would be the end of the strand of the molecule. And along the y-axis, we have essentially the vulnerability of that molecule to some kind of external chemical modifying agent. So along this chart, along this graph, the higher the peak, the more vulnerable the RNA at that position in the molecule. So if we look at the RNA with sodium only, we see one particular pattern of vulnerability along the molecule. If we add magnesium, we've changed the conformational state of the RNA, and we see a different pattern. Iron, we see almost exactly the same pattern, which is very interesting, what we'd expect based on the in silico results, right? So if we look at a different kinds of enzymatic reaction, we haven't talked about this yet, but we will tomorrow morning. Ribozymes. Ribozymes are like machines, like enzymes, like protein machines, in this case made of RNA, that can do interesting things like ligate strands of RNA, so they can reconnect two strands together to make one continuous backbone. So what we see in this reaction, if we test the ribozyme with iron in the absence of oxygen, and we subject that to time, what we see is that we get ligation product with magnesium, which is well documented in the literature. But in the absence of magnesium, but the presence of iron, we get a whole lot more ligation product, which is very cool. We can also take a ribozyme that does RNA backbone cleavage. So it's separating the RNA backbone. And over time, look at the fraction of cleavage product. We see that in the presence of magnesium, the ribozyme cleaves. In the presence of iron only, we get a whole lot more cleavage product. So overall, what this is telling us is that RNA, catalytic RNAs, can be much more efficient in the presence of iron than they can be in magnesium, as long as oxygen is absent, as long as we're recapitulating those early Earth conditions. So the very last and most recent experiment that we performed, this one's done by Chalong Shah of our group. Please check this out. This is a very, very cool paper. It was just accepted in Nature Chemistry. So what Chalong did is he took a colorless solution. This is tetromethylbenzidine. Okay, thank you. This is essentially a color indicator. So if this tetramethylbenzidine is oxidized, it's going to turn blue. Very simple. So we take tetramethylbenzidine, which is here, hydrogen peroxide, which is here and here, put in the RNA, put in the iron, remove the oxygen, but not necessarily in that order, and we can monitor the rate of change of the color of the solution using a simple spectrophotometer. Very simple technique. So we did this, or Chalong did this, not me. So if we monitor this over time, 20 minutes, here's our solution, 6 micromolar iron, hydrogen peroxide, minus, this is supposed to say O2, we can't have oxygen as reaction. If we have no RNA in the solution, we have no color change. We have no electron transfer. We add ATP, 3 closely spaced phosphate groups, which we would expect to interact with the iron, right? We still see no electron transfer. We add single-stranded RNA oligomer, no electron transfer. If we get electron transfer, we're expecting an increase in the blue color, right, which is going to increase this way. Double-stranded DNA, nothing. Large viral genome, nothing. 23S ribosomal RNA from Thermus thermophilus. Voila, we have electron transfer. P4, P6 domain RNA, which is a well-characterized RNA, small RNA. We also have electron transfer. And tRNA, also very well characterized, electron transfer. Very cool. We should be jumping up and down right now. Yay, Chalong. So what is common among those 3 RNAs that the other single-stranded RNA, the double-stranded DNA that showed no reaction? What is so special about those? Well, one thing we know from looking at 3-dimensional crystal structures is that we have these special magnesium phosphate geometries going on here. tRNA is probably the exception to that. I don't know that one of the magnesium phosphate backbone geometries has been observed in tRNA, but we do know that magnesium is very important to tRNA. This reaction also has the same kinetics as what we call the Michaelis-Menten formalism, so it essentially complies with the traditional test for an enzyme, which is cool. It means it saturates itself. So if we limit the enzyme, we have plenty of substrate. At some point, we're going to have so much substrate that we're not going to get any more catalysis per unit of substrate added. So conclusions, the critical role of magnesium in RNA, if we put it into a hypothesized early Earth environment in the absence of oxygen, can be better served by iron. The relationship with iron and RNA provides a very needed link between the RNA world and the geologic record, and that RNA may have originally served the sole role, iron may have originally served the predominant role in the early life, and that was later replaced by the oxidation, and that this new RNA world, RNA with iron, opens up a new realm of catalytic functions that have not been previously characterized. I wanted to thank the real brains behind this, not me, Chalong Shah, Anton Petrov, Dr. Shreyas, Denise Okafor, graduate student, and of course our wonderful PI, Lauren Williams, who's always there for us, as well as Steve Harvey, Nick Hudd, Dr. Wortel, and many others of our lab. And this is our lab, Lauren Williams Lab at Georgia Tech. Thank you.
All right, we can take one quick question.
Uh-oh.
I saw that you had the sodium control for your putting RNA with different ions. Have you tried any other divalent controls, like biologically relevant, like calcium or manganese, or?
No, we have not. One thing we do know is when we add magnesium to the electron transfer reaction, it kills it. So even if we have iron there with RNA, if we have magnesium there, it's gone. But we have not tried other cations, and we don't have reason based on our in silico results to believe that those would actually support any kind of catalysis. That's a great question. Manganese would be my first.
Manganese is relevant to the guy that was at
Mm-hmm. Yes.
Right, let's all thank Jess.